This is where we left off. It actually is on page 988 in your in the 11th edition. It's in other editions too. You just have to figure out what the page is. But one of the reasons why we are looking at this is there is something called the glomerular filtration rate. And if you've ever had a physical, which I'm sure you have, they have looked at this. They have looked at glomerular filtration rate. It is a mark of how well the kidney is functioning. And so that it actually is measured in milliliters per minute. So the average is about 125 milliliters per minute, which is really quite a bit when you think about it. That's about 10% of the fluid that enters into the kidney, the glomerulus, goes into the capsular space. There's also another, actually the more common test is the creatine clearance test, which I'm sure you've had. That's looking at a specific solute that is found in the filtrate and its concentration. If it's found in proper concentration ratios, then one assumes that the filtration rate is adequate. There are several things that can happen if the filtration rate is inadequate, and the next few slides will talk about that. So the factors affecting filtration rate here on this slide is the pressure of the fluid wanting to move out of those fenestrated slits in the capillary beds into the capsular space, minus the pressure back from the capillary space. So you have what then is the net filtration pressure. You have the capsular hydrostatic pressure, which is pushing back. You have the glomerular hydrostatic pressure that is pushing the filtrate out, but you also have pressure from the capsule as the filtrate fills that's pushing back, and you have to subtract those one from the other, the capsular from the glomerular, to come up with a net filtration pressure. The other thing that is going to cause pressure, and part of this net filtration pressure, is the fact that those solutes are going to want to, those solutes that are in solution, those colloids, are going to want to draw out based on a concentration gradient. So the regulation of GFR can be, so glomerular filtration rate can be local with just constriction. So it becomes one of the first things that happen is just constricting the arteries. If you constrict, you're going to slow the rate with which blood is going in the glomerulus, or if you dilate, you're going to speed up the rate with which blood goes into the glomerulus. There's also hormonal control. So there is autoregulation, that's at the local level. There's hormonal control, and there's also nerve control, autonomic regulation. So local, and then the hormonal control is this angiotensin, renin-angiotensin system. So if you have decreased blood flow that's coming into the glomerulus, that, remember, comes in the afferent arteriole. That afferent arteriole is surrounded by cells, which we call the juxtamedullary complex. And it has receptors in it that are going to pick up levels of volume. If the levels of volume are considered low, then renin will be released. Renin is a hormone that acts on angiotensin tensogen. Important that I say the gin part, because remember that anytime you see the gin, it's not active, it's inactive, but it can give rise to. So renin can turn angiotensinogen into angiotensin 1. It isn't ready to do its job yet. It needs to circulate to the lungs, where you have angiotensin converting enzymes, which we call ACE enzymes, that convert angiotensin 1 to angiotensin 2. And that then becomes a powerful vasoconstrictor. So that can actually act on these beds, vasoconstrict. It also tends to tell the adrenal cortex of the adrenal cortex of the adrenal gland. This is the gland that's sitting atop the kidney to release aldosterone. Aldosterone is a main regulator of sodium, and generally the thing that we say somewhere along teaching about how urines form is water follows sodium. So remember, our problem is we had decreased blood flow. So if we actually retain sodium because of aldosterone, water is going to follow it. So we are going to increase blood volume because of that. So this is a hormonal path of regulation. And then also remember, you have the autonomic regulation too by the sympathetic division of the autonomic nervous system. This image comes from your textbook. I tend to like these. These are they tend to be concept maps. After you have studied this, this might help you get the sequence down. So local response, if you have increased glomerular blood pressure, so if it's insufficient, homeostasis is restored. If homeostasis drops below normal, so if we're starting at the bottom right, so you have your bluish kind of circle, and then and that's when it's normal, so everything's good. But if you have decreased glomerular filtration rate, and that's starting at the bottom right, what's going to happen is you're going to have dilation of the afferent arteriole. Those afferent arterioles have the ability to dilate. You're also going to have a contraction of the mesangial cells. Remember those cells that angiotensin acts on to cause the contractile proteins there to contract. And then with that dilation, then you're going to have increased glomerular pressure, and then it's going to be restored. Now, if you have constriction, you're going to have it's going to have to go the other way. So dilation, increased glomerular pressure, restored. So too much volume of blood, the arterioles are going to constrict. It's going to be the opposite of this, but not enough decreased filtration. Everything, all of the fluid going in has to be increased, you think. So if glomerular filtration rate decreases, all the fluid going in needs to be increased. If glomerular filtration rate is too high, all the fluids going in need to be decreased. That's the local autoregulation. And then the angiotensin, renin-angiotensin system, what you have, again, you're going to start where you start with a decrease. So you have a decrease of filtration, so responds to a reduction in glomerular filtration rate. The endocrine response is renin is triggered. So renin in the bloodstream triggers the formation of angiotensin 1 from angiotensinogen, which is activated then to angiotensin 2 by the angiotensin converting enzyme, ACE enzyme, in the lungs. It then can do various different things. It can actually constrict arterioles, and if that's enough, then homeostasis will be restored. Or if that isn't enough, it can trigger the release of aldosterone from the adrenal glands. Aldosterone causes you to retain sodium, and water follows sodium. It also is going to cause you to drink more because it's going to increase your thirst response. And it's going to cause the constriction of venous reservoirs. So it's also going to cause increased cardiac output, and the two together are going to activate the nervous system to stimulate peripheral vasoconstriction. That would be a more severe drop, and then blood pressure is going to increase. The third thing that can happen with angiotensin II is it can trigger a neural response. So that can, as I said, stimulate thirst centers. The increase of another hormone called antidiuretic hormone production, which causes you to, and particularly that capillary, that collecting tube, the last portion that I said was permeable, sometimes more permeable to water than others, can cause that to be less permeable to water, so you'll actually retain fluids. Again, I think after you study it, this particular concept map becomes becomes more clear, I guess, in terms of what's going on. And then moving on to the proximal convoluted tubules, so the tubules themselves, where that's filtration and controlling the amount of filtrate that is produced. After the filtrate is produced, it goes into this tube system. The portion of the tube that's closest to the glomerular corpuscle is the PCT, or proximal convoluted tubule. The loop that's hanging down is the nephron loop, or the loop of Henle. And then the portion of the tube that's furthest away from the glomerular capsule is the distal convoluted tubule, which is the DCT. And then urine is formed, more or less formed. It just is a matter of whether it's concentrated more so, and sent to the collecting duct. Remember, this collecting duct serves numerous nephrons into the papillary duct delivered to the calyces. So the fluid moves along. About 99% of the water and many useful solutes are reabsorbed and turned into blood. The water is just used as a vehicle to move these solutes in and out, depending on whether they're needed, but mostly reabsorbed. The capillary beds that are found around this tube, so the paratubular capillary beds, reabsorb the materials, so reabsorb several materials: water, glucose, amino acids, hydrogen, potassium ions, and creatine. Some reabsorption is passive, but most is active. Most reabsorption occurs in the PCT, which we will talk about in the next one. I think I'm mispronouncing it. Creatinine. Sorry, guys. I think I'm mispronouncing that. In terms of the PCT and the reabsorption, this happens in various different ways. And this image is a little bit blurry. I'm sorry to say that, but I couldn't find it particularly in a clear way. And so you have it here, but you also have it in your textbook. And it's on page 993 in the 11th edition. And what's happening is each portion of the collecting tube is made up of epithelial cells. Remember, epithelial cells is your tissue that can actually secrete or absorb. So these are absorbent cells. And what can happen is various things can be reabsorbed from the filtrate, and various things can be not absorbed, I guess you might say. So you have water being reabsorbed, and you have that the farthest to the right, you have water and carbon dioxide being reabsorbed. Carbon dioxide is a byproduct of the buffer system, the hydrogen and hydroxy ion. The hydrogen in the